Yes. Sir, please. Good morning, friends.
Very good morning,
Good
sir.
morning to everyone.
Good morning, sir.
Okay, good morning. Shall we start today's session? Okay.
Yes, sir, I can start.
Thank you. Thank you very much.
Yeah, okay, sir.
It gives me a great pleasure to announce today's speaker in this session. Professor N.B. Ramachandra is well-known genetist in general in India and in particular in South India. He loves to teaching and he is very punctual about timing. See, exactly around sharp 10 o'clock we are starting. And he is one of the well-experienced researcher as well as administrator. So with the permission of Professor N.B. Ramachandra sir, because more than 47 pages of biodata is there, but I have briefed to 2 pages. If any omissions are there, please excuse me, sir. So I will briefly read out Professor N.B. Ramachandra sir profile. Professor N.B. Ramachandra sir is founder chairman of Department of Studies in Genetics and Genomics. It is offshoot of Department of Zoology. It is established in University of Mysore, Mysore. Presently, he is working as a UGC-BSR faculty fellow, Department of Studies in Genetics and Genomics. And he is also professor, visiting professor for Sri Satya Sai University of Human Excellence, Kalburgi. He is a consultant Human Genetics Unit, All India Institute of Speech and Hearing, Mysore. And also he is working as chairman, Human Ethical Committee, Mysore Medical College and Research Institute, Mysore. So he is a former chairman and contributed a lot for the growth and development of genetics department in University of Mysore. He did his a master's degree in zoology in 1982 and obtained first class with the second rank to University of Mysore. He has been awarded so many research fellowships. So just I will read out one by one. In 1982 to 1986, he has been awarded JRF Junior Research and SRF from UGC. Later, he got Senior Research Fellowship in 1986 to 1988. Then in 1988, he obtained his PhD from University of Mysore under the one of the great genetist, Professor H.A. Ranganath. Sir, PhD thesis title is Contributions to Public Genetics of Studies on Interracial Hybridization and B chromosomes. Sir also worked as research associate in 1988 to 1989 at Nimans, Bangalore. He is also worked as a CSIR research associate in 1989 to 1990 in the Department of Zoology, University of Mysore. Later, he went to abroad for pursuing higher research, that is in the name of a postdoctoral fellow, to McMaster University, Hamilton, Canada, where he did work on molecular genetics in the Department of Biochemistry. So, sir, area of specialization is Drosophila genetics, evolution, human genetics, diseases, and genomics. To attain excellence in these fields, he underwent so many training programs and learned from prestigious institutions like Indian Institute of Science in 1990. He has attended the genetic engineering professional course, and also he went to abroad, that is in McMaster University, and did postdoctoral fellow and isolated or cloned 10 mammalian HSP60 pseudogenes. He has also went to Los Angeles, USA in University of California, that is the Department of Cell Molecular and Developmental Biology, as a senior research associate. He has cloned 10 genes involved in brain development in flatworms. Of course, sir has visited so many foreign countries, and he has been invited to teach genetics and development in Department of Zoology, University of Innsbruck, Austria. He is also consultant for what I said previously, All India Institute of Speech and Hearing, course coordinator. Sir has developed two online courses, massive open online courses. One is human genetics and the second one is principles of genetics for undergraduate students in life sciences, MHRD Consortium for Educational Communication, New Delhi through EMMRC, University of Mysore.
Great, yeah.
He is a coordinator, worked as coordinator, NAC committee for preparation of University of Mysore in 2019. He has been also invited to select Indian Fellow of Indian Academy of Sciences in 2019-2022. So sir has completed almost more than 17 research projects worth rupees more than I think comes around 5 crores from various funding agencies, prestigious funding agencies from India and abroad. Sir has guided more than 25 research. Please mute your
Aruna madam.
Yeah, please.
Mic is on.
Yeah.
Sorry.
It's okay. And he has presently around more than 3 students are working under his guidance, and 2 MPhil candidates successfully completed their course under the guidance of Professor N.B. Ramachandra sir. Very interesting thing is, so under the guidance of Professor N.B. Ramachandra sir, so about 9 students worked as summer students, summer fellowship under the summer fellowship sponsored by Indian Academy of Sciences. So about more than 273 publications published in very reputed journals, both in foreign and national, and peer-reviewed journals. And sir has deposited more than 85 gene or DNA sequences in NCBI USA. And also he has contributed research notes in Drosophila Information Service, United States of America. So he has developed more than 5 protocols and methods and 14 books and documentations, and attended 249 seminars and presented in all those seminars his research findings. Sir, H index is 24, I-10 index is 60, and the total citations are more than 2000. And sir is also known in this campus and South India and in India for academic, administrative and professional responsibilities held at various occasions. With this brief introduction, I welcome wholeheartedly on behalf of my personal behalf, on behalf of all the teachers and on behalf of academic staff's college to this session. I welcome you, sir. Please over to sir. Now sir is going to take up the lecture. Thank you.
Good morning.
Good morning, sir.
Thank you, Dr. Basurajappa, for very lengthy introductions.
Thank you, sir. Thank you.
Well, if anybody is interested in my field, you can visit my own website, ramachandralab.com. It's fine. Now, I don't know, some of you might have attended this now your genome classes in different places. I gave in Goa also. I gave the almost same, last month, yes, there was a refresher course. Anyway, so I want to introduce the genomics and genomes, our own, and what we know so far. And I want you to have an interaction. You can stop me anytime. You can raise your hand. You can stop me anytime. We'll have a discussion. Okay. Let me start now. The content of my talk deals with 5 subheading: genomes, genomic variations, and a few examples of our own study, and how genomes have evolved and where it stands in the genomics today in global. I believe you know what is genome. Genome is nothing but the entire DNA of an organism, including genes, sometimes even the extra nuclear DNA also includes a genome, mitochondria, as well as chloroplast. And this genomes were in higher organisms bifurcated into chromosomes and all put together, all the chromosomes, mitochondria, chloroplast, all together, it's called as a genome. You know, the chromosome has the genes and so on. Many organisms are the genetic material is DNA, but however, there are the viruses are coded by RNA, and the RNA, we call them as RNA genome there. So when you say a genome is not only coding sequences, it also includes non-coding sequence of the DNA. I hope all of you are aware of this. Then why we should study the genome? What is the necessity of studying the genome? So each and every one of us is unique creations, and we share the same ATGC, microbes and human and animals and plants. And the hidden code determines which protein should be made and when. And the language, as I said, is common for all organisms. It is now in the 70s and 80s, there was a very revolutionizing of mixing of the DNA of plants or microbes or the animals. That is for a revolution of manipulation of DNA. Now we are in the verge of, or we are in the new line of genome editing. So the genome sequencing has started in 1990. I'll come to that little later. So we learned from the bacterial genome, and that will stimulated the contemporary scientists to go for the human genome and other contemporary. What do you learn from the bacterial genomes? So bacteria maintain the very good economy of the genes, the coding genes, and there will be very less of the non-coding sequences in the bacteria. When you look into the genomes of the bacteria, different bacteria, the genetic diversity is greater than what we thought. And as most of us know, mutation is the major source of the genetic variations. And non-coding arrangements of functional elements like cistron or the regulatory genes, or one regulatory elements control a couple of genes. I hope you can remember lactoperon and other operon systems. So generally in bacteria, you can consider 1,000 nucleotide constitute a gene. Bacteria less than around 1,000 nucleotide. As I told you, there's a wastage of or the non-coding sequence is almost zero, or if it is there, some about 100 nucleotides. So when you look at the quantification of the genomes of different organisms, starting from in a eukaryotic system, and this is the x-axis and y-axis, we have the organisms as well as the size of the genome. If you look into that very broadly, algae and protozoans have the wide range of genome size. You see the amoeba, it's a small unicellular organism, but the genome size is vast. It's not been sequenced now. And algae also is large. And insects also varies from the different kinds of insects. As you move the hierarchy, the complexity of organism, the birds and the mammals, look at the range of the genome size is minimum, and the mammals, including mouse, elephant, and human, whatever they belongs to this mammalian system. Whereas flowering plants, we do have again the wide range of the genome size variations in the flowering plants. And these genome size variations in the 70s and 80s, people are thinking of C-value paradox. What could be the reason for this kind of variations? Now, because of genomics and the advent of molecular techniques, we could able to resolve these issues. So, for example, genome size does not correlate with organism complexity. right? For example, amoeba, if you look at the genome size of amoeba, 67 billion base pairs, human around 3 billion base pairs, yeast 12 billion base pairs, and having 6,000 genes, 2 kb per gene in that sense there. Okay, so humans are the more complex organism, highly evolved in the nervous system, and they have less genome size, whereas amoeba, that's why the genome size does not correlate with the organism complexity. And the why plants have wide genomic diversity, as we already know, plants are not the haploid only or diploid only, even they are polyploid plants. And Barbara Beckleton studied for 40 years, and she invented the transposon jumping genes in plants. They're also responsible. Later studies also identified gene duplications or more in plants, not only polyploidization, transposition. These are responsible for variation or expand the plant genome size. Eukaryotic genomes are filled with large tracts of non-coding, often repetitive DNA sequences, and only about 1.5 to 3% is coding for the coding genes and non-coding genes in the human. Remaining things is a repetitive DNA sequences. Earlier used to call as a junk DNA. I don't think now we are allowed to call this a junk DNA because we do not know the function of this DNA. To very briefly to tell about our own genome, the Human Genome Project has started in 1990 with great ambitions and with a lot of the oppositions, and they planned it for 15 years but completed in 13 years, and they coordinated by the US DOE of Atomic Energy and Department of Energy and NIH, and later 18 countries are joined together to complete this process in 13 years. Later, last 3 years, the private funded, the cellular genomics also joined in hand to complete the Human Genome Project of the sequencing within 13 years. The objectives, as most of you are aware of this, to Genome Project, to identify, determine, store, and develop the methods to know the number of genes and how to store them and how to retrieve them, and also after knowing the genes and the genome, also address the ethical, legal, and social issues. And this has been in 2001, compiled working draft of the human genome has started. The public funded has compiled it and published in Nature Journal in February 15th of 2001. And the private funded cellular journal is published in Science 2016, and working draft of that. Later, it took about 2 years to compilations or completion of human genome. That is April 14, 2003, they announced the completion of human genome, which coincides the 50 years of the elucidation of double helical structure. I hope all of you are aware of this. Then now let's, so knowing the genome, Human Genome Project, then what is genomics? Genomics is study of the genome by using analytical method and asking scientific questions and set of techniques to understand, unravel what is lying in the complete genome. And the steps involved in the public funded one, there's a very step-by-step, the one isolation of the individual chromosome, fragment them, and correlate with the genetic map and develop the physical map, a fragment and cloning into a smaller, larger vector, a smaller vector, and sequence them and arrange them also together. That is the bottom-up method there. Whereas the private funded, they did a shotgun sequencing method, they could able to complete that in 3 years also. So the message from the whole genome sequencing is that the coding, protein coding sequence is around 1.5%, and the line sequences, I hope you know what is a line. long interspersed nucleotide elements, and like sign is short interspersed nucleotide elements. Lines are about 6.5 kb. They are like transposons. Human L1 transposons are under this line sequences, and about 26% is the intronic sequence, which is not coding for the particular protein, and many are the repetitive DNA, as you can see, we could able to see in our genome. So our genome is more of repetitive DNA and non-coding DNA, only coding DNA for the protein, as well as the other RNAs, the coding RNAs are there about 3% and so. Generally, when I say the humans are complex, when compared before going for the human genome studies, people thought that Drosophila is a smaller organism, might have 10,000 genes, whereas the human, if you compare that 1 lakh genes, but it's not so. The human have only about around 25,000 genes. Now people coming up with that 21,000 genes, protein coding genes. Rest of others are the non-coding genes in the human. So it's the 20 to 25,000 genes, depending on what kind of the analysis you are going to make. But the 25,000 gene proteins are making our life normal, or all our metabolic activity, all our normal functioning, and each protein has more functional properties, a tissue specific property. That is the one is known. After sequencing not only human genome project at 20 individuals, there were later from millions of individual sequence have been sequenced, and all the human genome sequence have been analyzed. They made the comparative account analysis they have made, and they identified the differences in the annotation and the comparative account of the similarity differences, and that will tell us about. Generally, we have about 99.6%. We are all identical. All the human beings are identical. Only 0.4% is differences we see. This 5.4% differences is the 4 million bases are the one making us different from each other. And the information, whatever generated from the genomics is affect the society in many ways. After completion of the human genome project, many technology developed, many kits have been developed. American government has spent about $4 billion, around $4 billion for the human genome project. Later, they earned nearly about $800 billion, as economy is improved because of the genomics and the other techniques and the methodology and the instruments, whatever they develop. And that is the economy has increased, the American economy has increased, and now the UK economy is also with the genomics is going to be increased in this sense. To briefly tell about the genes and the genome size, the chromosome, the chromosome number 1 in human is the longest one. It's having 279 MB of sequences, but it has around 3,000 genes. And if you go down here, the general is based on the size the chromosome have been arranged. When you look at the chromosome and look at the chromosome 8 and 11. 11 is the smaller than the 8th chromosome, but however the gene is double, almost like a double the number of genes present on the 11th chromosome compared to the 8th chromosome. It means that the gene number and the size of the chromosome is not going together. Similarly, another example we can see here, look at the 18th chromosome and 19th chromosome and the 17th chromosome. 19th chromosome is smaller than the 18th chromosome. However, 19th chromosome is in the size is smaller, 72 MB, but it's more double the number of the genes it harbors also. So I hope you know that the culprit chromosome is the for the Down syndrome 21st chromosome, it has around 300 genes, and the X chromosome where the female has 2 X chromosomes, they have 1100 genes. Y chromosome we have about 240 231 genes in the Y chromosome, whereas male are less genes compared to the female because the female have 2 X chromosome. The thousands of genes are more in the female there. If you compare the complete the genome analysis, like we have the female have 23 linkage groups, the chromosomes pairs, whereas males have 22 pairs of autosome and X and Y chromosome, and the length of the chromosome varies from about 25 folds. First chromosome, the last chromosome, 25 folds smaller, the last one, chromosome. The organelle also had the genome, and the relative low density, 50 genes per million base of DNA. That's it's low. Only 50 genes are present per 1 million base pairs, and the length of one cell DNA to unbind all the 46 chromosomes, it comes to about the 2 meters, about 6 feet. So length, and how many cells we have? How many cells we have? We are all developed from a single zygote. How many cells we have? Many, right? Billion, trillions. How many? Can anybody tell us some quantification? Computer biologists will quantify them also. I have not mentioned here, but I have the answer that if you anybody knows, you tell me, otherwise I'll tell you. You add, it is 10 to the power of 13 to 14 cells we have an adult human. That is, for the in front of the 10, add 13 zeros or 14 zeros, that many cells an adult human being has.
Around 37 trillion.
Yes, that is the 13 to 14 folds of the 10. That is 10 to the power of 13 to 14 folds there. Okay, so trillions of cells we have, and each cell has the DNA except the matured RBC. And also, if you look at the still integracy, muscles are multinucleated cells, the livers are polynucleated. Now you come to know that if you sequence the liver cells and the muscle cells and the other RBC cells, if you take the only blood cells, and the differences we can see in the genome there. Okay, we do see those kind of. Which is the smallest gene we found in our genome? Any idea? Smallest gene and largest gene. Because now my title of the talk is the our own genome, knowing our own genome.
I think largest gene is dystrophin.
Excellent. Yes. Smallest.
Smallest.
Well, the largest, the gene size ranges, including non-coding genes also, 21 nucleotide to 2400 kb. This 2400 kb is the dystrophin, and 21 nucleotide is a microRNA. Okay, and tRNAs and RNA are having about 65 to 75 base. And the smallest, the protein coding genes is the insulin-like growth factor, is having 252 base and 67 amino acids. As somebody told that the longest gene is the dystrophin with 8 introns and only 3,685 amino acids. Rest of is the non-coding, that is intronic sequences in the one. The dystrophin easily one can remember. One human dystrophin gene is equivalent to half the bacteria Escherichia coli genome. Escherichia coli is having 4 MB, right? This is 2.5 2.4 MB here, the dystrophin itself. But it's coding for only one protein. So that is the kind of diversity we have the gene. Another interesting one is that tritine having the exon, the one single polypeptide of carrying continuous 27,000 amino acids. It's a unique feature. And more dense genes are seen in the 19th chromosome, that is 23 genes per million base pairs in the 19th chromosome, which is depending on the size and compared to all other chromosomes, 19th chromosome having more the genes there, as already I told you there. So only 5 genes are seen in the 13th chromosome. Here the 23 genes is almost like 5 times more than the 13th chromosome there. And we do have very specific the proteins for the human, that is antibodies and T-cell receptors for antigen, and the HLA, that is major histocompatibility complex, is very specific, and the cytokines are very specific to human, the cell signaling molecule, and also blood typing factor are specific, and mediator of apoptosis, which is the programmed cell that is unique to the human. These are the very specialized, very unique to the human system compared to the other system. So after sequencing the human genome, taking the 20 individuals, anonymous individuals, and the blood and the sperms, blood from the female, blood, sperm from the male, mixing them, making a cocktail and distributing the 18 countries laboratory, and 30 working on 13 years, they made the cocktail genome. And finally, somebody asked that we cannot compare the cocktail genome with any one of our ethical groups there. So 2000 itself, people have started asking questions. It's not going to resolve any issues. So let's have the ethnic specific genome sequence. So post-genomic era, people have, once the technology is available, the instruments are available, and the scientists were available, they are in a race to sequence as many as individual also. In 2007, Watson, Craig Venter, their own genome has been sequenced by the company there. So after the genome project completed, even the working draft completed, many new post-genomic projects have launched. International HapMap project has started, 270 individuals belonging to 5 ethnic groups. Individual genome project has started, 1,000 genome has started, and ENCODE for the regulatory sequence of all the genes have been started. Synthetic genomics and personalized genomics later, it has started in the 2012 onwards. And 1 lakh genome project, several 1 lakh genome project, 1 lakh genome project of Asian 1 lakh genome project. The UK 1 lakh genome project has started, and now in India, a 20,000 rare disease genome project has started in 2019 and so on. So many countries have started the whole genome project. China has even 6-7 years, they plan to sequence every individual, very newborn babies, and also UK is now leading with the genome sequencing of the patients there. And later, with the advent of technology, genome editing is possible. Genome editing has been successful in many plant and microbial industry, even in human industry also. That is ethically, it's not allowed to do that. So after reviewing the genome and genome projects very briefly, and what make the difference between us. So because we are discussing about our own genome, why I'm different, or how I am different from my sib parents, or am I 100% identical? No, you are not 100% identical. You are 99.6% identical, even your parents, as well as your brother or sister, or any other your neighbor or the friend in the ethnic group. What are the reasons for this kind of the changes or differences that they have been called as genomic variations. Look at these two individuals. I hope you might have seen this. They are, do you think that they are normal or they are abnormal?
Abnormal.
Abnormal, sir.
How many will say normal? None? I say they are normal. Normal. Why they are abnormal? Or why they are normal? How can we explain this? This is because of genomic variations. Both are normal, they are living like us, and the height gene has shown the genomic variation that makes them to, along with that, okay. Before going for the complete genetics and genomics, every individual has the genetic material and also their own environment. So for any kind of our characteristics or any kind of our phenotype features, and both 50% is the environment, 50% of the genetic material are responsible. Don't always think that only genetics play a major role or genetic material play a major role. Even environment equally play a major role to express those genes or suppress those genes, even though if they're defective, the environment makes them. For example, now the hypertension or the diabetes, you look at that. The diabetes, doctors advise not to eat more sweets or not to eat sweets at all. Either eat sweet, more accumulation of the sugar. So that way they can manage this. People are lived for 100 years with diabetes. People not able to live even after 30 years because they are not maintaining their environment properly. That's one thing. And again, the genes responsible. So for any characteristic feature, any phenotype, we have to consider the environment equally and as well as the genetics. So here, the genetics factor here, the genomic variations in the height genes are responsible for the differences. Otherwise, they are normal like us because I can say a couple of 3 or 4 members here, and even when you compare with me, my phenotype, my face is different from your face and other face also. That is because of the genomic variations. So, how do you quantify the genomic variations? The genomic variations is nothing but changes in the genome sequence between individuals are called genomic variation. Major causes of genomic variations for any is the, as you know, some mutations, which is the raw material for evolution, and also in a normal activity, sexual reproduction, where in sexual reproduction, you know, the meiosis, the parental, the genome undergo recombinations during meiosis, and also homologous recombination, and also independent assortment of the chromosome takes place that also contribute for variation. And during fertilization of 2 gametes, unique sperm and egg, that also contribute to genomic variation. So mutations and sexual reproductions contribute the genomic variation. Whatever we have differences between us is because of the naturally occurring the spontaneous mutation as well as the sexual reproduction events there. I hope you believe. Do anybody has any other example or any other the suggestion or comment? The major causes of genomic variations are the mutations and the recombinations during meiosis and fertilizations. Meiosis, when the gametes are produced, they undergo recombination, produce a unique gametes. And also these 2 gametes, during fertilization undergo recombination, undergo the fusion. There also the dominant, recessive, or interactions, all such kind of things happening there that also contribute to variation. Okay. And who is normal now? Our Modi ji is normal. Very interested person, like the model, your parents, or any of your persons are the normal. Who is normal here? So how do you define the normal? When I was giving a talk in IASA, then we had about 10-minute discussion about to how do you quantify, how do you say it's a normal? When you say normal, most frequent, we are all normal. How? 99.6% of the genome is normal. So we are normal. Only 0.4% is we are different. Okay. So depending on how much identity of the nucleotides are there, that based on that, we call this a normal or the abnormal. So another term you should know, the polymorphism. The single nucleotide polymorphism, generally called as SNPs or the SMP. I hope you heard, most of you heard about this. So these are again the mutations, again the variation. They are more frequent variation. How frequent? 1 out of 100. They call this a polymorphism, 1 out of 100. 100 nucleotide, 1 nucleotide is a polymorphic or is not having, when you compare two individuals, out of 100 nucleotide, 1 nucleotide is not the A, it might be C or G or T. Okay, so that way you quantify. Earlier they say the SMP are not causing any kind of effect on the phenotype. Later, with the advent of the technology genomics, we do assign a particular SMP is become a causative agent or factor for causing the variations or the diseases and drug resistance and so on, or susceptibility and so on. And the other term is mutation. What is mutation? What is spontaneous mutation? What is the rate of spontaneous mutations? Any change, any heritage. Generally, when you ask the student, what is mutation, they say sudden change in the nucleotide is a mutation. It's not a sudden change, it is a heritable change, which is the change occurred in the gametes and pass on to the next generation is heritable. And then you can ask the question that if the cancer is a somatic, they call somatic mutation. Generally they call this a heritable change, the genetic mutation. The one passes generation to generation, where somatic mutations occurs in our somatic tissues and cells. They may not be heritable, depending on if the mutation, if the changes occur in our germ cells and so on. So mutations are the very, very rare. It has been quantified long back, 40 years back, 1 in 1 million to 10 million. After 10 million or 1 million nucleotide, only one changes are there. That is a mutation. That's why the mutation is a gradual process of the evolution of the organism and so on. So mutations are very rare. Polymorphisms are very frequent. Okay. So now the normal, depending on how much we have identical, then we call that a normal. I hope I cleared your doubt, or did I created doubts in you? Is it clear?
Yes, sir.
Thank you. So classification mutations. I hope you know this. I'll go rush into this. Causes by spontaneous and induced by chemical radiations, and even the biological, like the transposons can be used. We can induce mutations, and it can occur somatic cell, germ cell, or any region of the genome mutation can occur. The site functions, it can develop reverted mutation, lethal mutation. Loss of function means the protein, if this mutation, it lost the functions, and the gain of function, there was a stop codon, the stop codon converted into some amino acid, then it's the gain of function, it will be there, or in the promoter region, the is defective, and the promoter region, the gain of function, it can cause the protein to be active. Fitness, it can be some mutations are neutral, beneficial, harmful, depending on the environment, what environment you will ask them. And size of the mutation from single nucleotide to segment of the chromosome or the whole chromosome, broadly it can cause this mutation. So if you have any clarification, discussion, you can stop me. Okay. Why we have to study the genomic variations now? To understand which variation is neutral, which variation is pathogenic, which variation is not going to cause any problem for me at this age or the later age or in a young age also. So knowing about this variation is very important for the normal survival of the individuals. And also by looking into the genomic variation, we can also look at the size of the variation, location where exactly these variations are present, and how much this will make the impact on the phenotype also very important. So pathogenic, neutral, the size and the location, all these is going to be helpful to understand what kind of phenotype it can produce. So even this understanding genomic variation, we can trace the human migrations, expansion and adaptation. And also now last year Nobel Prize is given for the guy who has identified the sequence and compared the sequence Neanderthal of the human and with the genomic sequence gave a better evidences of how close we are, how different the ancestor to the Homo sapiens. So broadly, genomic variations are classified in 2006 into 4 different categories. First is the SMP. I hope you know this. I already told you, single nucleotide polymorphism, 1 nucleotide change. And next comes the microsatellites. I believe you know the microsatellites. When 2 to 10 nucleotides are added or deleted in a genome, and they are called, I hope, in a say microsatellite, like an essential DNA profiling, the microsatellite may be present in the coding region or non-coding region. They do play a major role for the genetic diversity. The third category is the indels, small indels, insertion and deletion from 100 nucleotide to 1000 nucleotide. Any variations or any sequence are inserted in the genome or deleted in the genome, they are called as indels. And 1 kb to the 5 MB, million base pairs, the insertion or deletions to our genome or duplication or deletion genome, they have been called as copy number variations. I repeat again, we have 4 different kinds of genomic variation: SMP, microsatellites, indels, and copy number variation, that is CNV. CNV harbors major chain. If you look at the genome sequence of the Craig Venter, his genome size is smaller than an average human being, 2.8 billion base pair, whereas normal we say 3.2 billion base pairs. Because he has found many the copy number variation, deletions in his genome also. We'll discuss a little bit about that later. I hope we are clear about this. And later, we also know the chromosomal aberrations like deletion, duplications, inversions, and translocation. These are the classical one, what we teach to the student. Now, these genomic variations are, with this genomic era, we have to teach, we have to understand more about these, because these are the one causing more pathogenic situation, or some of them are there, the people will be afraid, and you can tell that they are not going to cause anything because they are neutral. Understanding more of genomic variation at this stage is very, very important. Especially people like you, not like me. I hope you are at least 20 years younger than me, some of you, and you should know more about that also in this situation. The genomic variations can occur anywhere in the genome. They can occur in the coding, the non-coding, that is regulatory sequences, or within the change in the amino acid here, or in within the sequences, or become the nonsense, or in the splicing site, or intronic or intergenic regions. So exonic, intronic variations are also very, very important. If the variations found in the exon intronic region, then it will be a problem. Yeah. How much time I have? Another 20 minutes? Right? Yeah. So let me take about the microsatellites. As you know, the microsatellites are repetitive sequences of the CAG is a repeated sequence, Huntington. If this CAG repeats are up to 35%, they are normal. In our normal, if we are normal, then we can have up to 35% of CAG repeats. If it is more than 37 or 100, then they are Huntington's. Similarly, Fragile X or the myotonic dystrophy, the repeated sequences, the trinucleotide repeats, if they are more, then end up with the Fragile X is become as a mentally retarded. If they have more than 54 repeats, they become mentally retarded. As I told you, trinucleotide microsatellite can be present in the regulatory sequences. Then the Fragile X, they are present in CGG, they are present in the regulatory sequences, or spina bifida, muscular dystrophy, they are present in the coding sequences, or the muscular present in 3' UTR, Huntington, the coding sequences, they can present anyway. They affect the functional property of the protein. So single nucleotide polymorphism, as you know, as I already told you, now the scientists are with a lot of the information, lot of the data is available, big data is available, genomic data, we could able to assign them for the drug resistance, drug susceptibility, and so on. And our own work for the asthma genetics, we could find that some of the asthma patients who are not happy, or the steroid what the doctors were giving, they are not helping them, and then we sequence them. We found that SMP is causing the resistance for them and so on. I'm not presenting that data, but it's there. Recently, we published that in the diagnostic with the JSS Medical College, Dr. Mahesh and so on. Nucleotide substitutions, we know transition and transversions. They create silent mutation, missense mutation, nonsense mutations, and they are very, very important. And this synonymous mutations, changing the same, nucleotide change is there, but amino acid not change. They may not have much effect, but still we can think about later the codon bias effect. That also causes, also if we look into the greater understanding about that. But non-synonymous, changing one amino acid to another amino acid. Classical example, we see the sickle cell anemia here. The glutamic acid change into valine, and that become oval shape to become sickle shape. One nucleotide chain make one amino acid leads to one amino acid chain. Whole lamina of the blood cells vary. That one mutation, one chain is sufficient for this. The third category is the indels. For example, the genetic code here, they create the missense or frame sheet, and the loss of any nucleotide added or deleted, and it create the frame sheet also. These are also very, very important. We should understand in our genome of this. And we did some of our studies from last 30 years, 20 years, 25 years also. We did publish on this indels and the SMP in many, like the congenital heart defects here. We found them, and asthma also. We found asthma. There's IL-4, RM33 gene polymorphism we found, and also in the insertion deletion we found in a Tibetan population and so on. So the third one is the major one, the copy number variation ranges, and they are the insertion or deletion or duplication, you can call them. It has more deleterious because you are losing about 500, the 5 MB sequences, several genes will be deleted, or if the several genes are duplicated, it will going to have drastic changes. The copy number variation is major, and from last 15 years, my contribution to the science is mainly in copy number variation using the 1000 genome project which is available using, I think I'm going to tell something about this. So I'm going to cover some of our aspects in this. Some of the work what we have done, one or two example, I'll take it here. So how the copy number variation occurs? Copy number is generally we need to have a diploid organism. We need to have 2 copies of the DNA or the gene, whatever the segment. Sometimes if you miss 1 copy and both the copy miss, will have 0 copy number, and sometimes 3 copy number, 4 copy number. This is because of the homologous repeats, segment, the non-allelic recombinations, the chromosome wear, all these are the mechanism involved to produce the copy number variations. They have been proved there. So if you look at the Craig Venter already told you 3 million SNPs are there, and about 20% of the SNPs, the single polymer, new, they are not found in the Human Genome Project also, and also he's sequencing his own genome. He found that he has a problem of cholesterol accumulating problem, and he started taking the statin. Even in 2007 sequence, 15 years ago, he started taking the statin to thinning the cholesterol and so on. So he can manage his own genome, his own by personalized medicine. A few examples of our work, maybe if your time permits, I'll take 2 or only 1 I'm going to explain. So I had very interesting PhD students, and they were, since we are doing the 1.2 million the markers for the screening of the asthma, dyslexia, and different kinds of diseases, and then also the publicly available database in the net. Databases we found that 1,715 individuals belong to the 12 population. The genome, the microarray analysis we downloaded. Our own sample is 43 samples and 270 of HAPMA samples and 31 Tibetan. They are a different one. Chinese sample, the Jewish sample, and this Taiwan sample, Australia, and New World. This is nothing but American samples. So all the samples are present, except ours is the present as the unprocessed data. We got it affirmative genome wide SMP 6.0 from the array express archives by European Bioinformatics Institute with the understanding of we got it permission from them, downloaded, we started analyzing them by using different genotyping platform, and we have these 2 kind of what this array. What do you mean with this array? They are the 1.8 million markers and 2.6 billion combined SMP single nucleotide polymorphism and copy number variation markers were there. And these samples, 1,715 samples, were using with this array. They are available, and we started using them, asking different kind of questions and screening the whole genome with this 2.6 million markers. So we used the different kind of algorithms, softwares for them. The publicly available softwares. Some of them are commercially. We registered for them. We got them, and the study here is that when you understand, analyze the copy number variation in the whole, in all the genome, complete 12 population genome, 1,715 genome, more of duplicates, 74% or 75% of the duplication we see, the copy number duplication rather than the deletion. Deletion only 25% in the genome. Systematically, we analyze this, the genome. We made about different questions. We asked nearly 18 research publication in the peer-reviewed journal. We made different kinds of one or two. I'm going to present it here. If you look at the genome size here, copy number, the more of the 100 to 100 or 250 KB deletions or duplications are more here compared to the 4 to 5 MB sequences, deletion or duplications. And okay, if you pool all the data of the 12 populations, I wanted to look at here India. 10% of our 43 samples, what we made the same array. And look at I want you to look at the China here, another China HapMap China sample 3%. And I want you to look at this here, HapMap YRI, these African samples. Here, African and China samples only 3% of variation. India has a 10%. Look at the New World, means American samples had the 16%. I want you to concentrate on this. Africa and China has less variations with these markers. India has the 10%. India, Australia, or Tibet, they have about 10-11%, and even German people also have about 9% coming to this category, whereas New World has more of variations. We mapped this. We found 44,109 copy number variations. We mapped individual all the 12 population for individual chromosome, and an average a gene will have about 1,329 copy number variations. It's a huge work and data of the 6 members. Other than the PhD work, they did it for 6 months. If you look at the population wise, I want you to concentrate light green and dark, light green and dark green in these cases. What is that light green and dark green? Duplications. The CNV present in the coding sequences, duplication and deletions here. Dark green is the duplications found. Wherever you see here, African population, the German population, Jewish population, here Jewish, and Tibet and India and Australia and Taiwan, we do have more of duplication in the coding sequences, whereas the deletions in the coding sequences we found more in the deletions in the China and as well as in the Japan population there. And a non-coding, this way we analyze the whole kind of genome with the coding and non-coding, all kind of questions we ask, and also we map them also the cellular involvement of the impact of the CNV and gene participate in cellular system like developmental process and response to stresses, signaling cascades, the biosynthetic process, phosphorylation, and different apoptosis. All they have been quantified. How many genes are there, and those genes are hit by the copy number variation in a globally. We classify them. We use copy number variation to establish the human migration. So as a marker. So people used to use our Raji Singh or our Tangaraja group did use the Y chromosome and mitochondrial DNA as a marker to trace the human one. We use the copy number variation and here, and as I already told you, this African have only 3%, and that was first settlement from Africa. The population, human population moved. A second settlement in the China, around China, again the same 3% of variation. From there, they distribute their spread, and then they moved to the America. In the New World, they have 16%. The variations are found. So this is in addition to the whatever they had, that the copy number variation can also use as a marker to understand. So we published this in the PLOS 1, and have been well received. The appreciations about this using this also. So we made a global map of the CNV map. It can be used, anybody we deposited, and to understand more about that. Using this CNV 44,000 CNV map, we asked different kind of, can we find the dyslexic specific gene? Do they hit with the copy number variation, either diabetes and so on? We did them, and we even Parkinson's disease also. Whoever my students have the interest, we analyze them. We could able to identify the genes response or gene are associated with the CNV, and they have to look into that also. We have found that. One of the another interesting case. How much time? Yeah, another. I think I'll take another 10 minutes. Less than one more one here. I want you to look at this pedigree here. Wherever we block here, the shaded, they are males, right? Grandfather, father, and two twins, right? All these four members had the dyslexia. So what do you think about that? What is the patterns of inheritance of the genes involved in this case? Easily you can say they are Y chromosome, because the grandfather contributed exclusively Y chromosome to the father of these two kids, and this father contributed the Y chromosome to this, right? Easy, but it is not so. We worked 2 years on this aspect. We found the copy number variations and the coding, non-coding regions, that is the regular regions and coding region duplicated in different individuals. To make the story short, we found there is one more regions between the X and Y chromosome undergo recombination. Y chromosomal segment move to the X chromosome, and mother, one of the X chromosome of the mother has this defective gene, the protocardian gene, which is present in the mother, but she's having two copies. But wherever this defective gene present in this segment, protocardian, all these four members have their dyslexia one. So protocardian is a cell communication the protein, and we worked out greater detail. We will publish this also. So what I want is here, original contribution here. People know that the terminal regions of the X and Y chromosomes are called as PAR1 and PAR2. Mean pseudo autosomal regions. They undergo recombination in 1980. Scientists have proved that. From our lab, we made another regions here, PAR3, where the recombination between the P regions of the Y chromosome and the Q regions of the X chromosome undergo recombination, and this has been proved in general population also. We made published. This is one of the contribution from the university, where we are struggling to get electricity and the drinking water, and we could able to because of my good students and our thinking, and we could able to make this. This has wide citations of this. Many people now accepting that a new region between this. Whatever the 5 genes are coding in this region, they exchange, and here frequently about 2% of recombination. You can see here we did this experiment. So any scientist here, every observation is unique and important for new discovery. So when we understand this, the students is not able to solve it, let us leave it. We pursued it for 2 years with different kinds of examples and the questions, whatever the reviewer asked, we could able to prove that there's a new region between X and Y chromosome here. Recombination region, the PAR3 is new to science. This is the contribution from University of Mysore. This is the way this many copy number variation we could able to see in not only 6 individuals of belong to this 3 families. We could able to see this, and we trace that, and it has been accepted. And several copy number variation we have reported and so on. So how I'll take 5 minutes also. How genomes have been evolved. First, the transition of genome evolution is the first eukaryotic system from bacteria around 5,000 genes, 1.4 billion base, 5,000 genes into 10,000 genes. First eukaryotic system duplicated double the size of the gene. The second transition, the first vertebrate, is about the Cambrian time, 530 million years ago, and that's from 10,000 to 30,000 genes, where generally we say the human and the mammals have 30,000 genes and so on. So this there are 2-way gene duplications. One is the new genes have been acquired by duplication of existing gene, or acquiring the genes from other species. These are the 2 examples I think is okay here. A gene is duplicated, and this gene is stays similar. The gene stays similar. Both the genes are working like ribosomal gene ribosomal RNA genes are thousands of copies are there. They all are needed to make the ribosomal RNA, to make the ribosome for protein synthesis in our the system there, in all the eukaryotic system. And one of the gene undergo selective pressure and degrade, and only one is functional. Other gene they acquire a new function and become a new one. So hemoglobin, myoglobin, they are the two genes that evolved in this fashion. So gene duplication helps to evolve new genes. And this is the way. Another concept, as you know, I think you taught transformation, transduction, conjugations also, right? This is the one we taught. A lateral gene transfer in eukaryotic system can occur from mitochondria to chloroplast, from chloroplast to nucleus, from nucleus to mitochondria. On all these lateral gene transfer, there are n number of examples we can see. Lateral gene transfer, gene duplication, or making the new evolution of the new gene or the new genomes there. So some of the scientific breakthroughs here, happiness gene means the regulator of the serotonin is very, very important. If it is not there, even though they give 10 lakhs per month for a salary for the UGC fellow, they are not happy. Even people, the guest faculty, they are getting 10,000 or 20,000, they are happy. Okay, because our serotonin has to be regulated, and the regulator gene have been identified, that has been called as happiness gene. And the sports gene, many times they will say that my son should get a Nobel, the Olympic medal, and his ACTN3 is not normal, fast muscle fiber forming gene, is not normal, he cannot make it. And this also now been identified. And if these genes are working, proteins are making normal, and we can train them. Again, environment, we have to provide them, right kind of environment. I have a lot of the short story on that. One businessman came, a 7 years old son, he wanted to make Olympiad, and we got sequence, and also we made advised him also in that cases. And crane gene here, monoamine oxidase here, if the low level monoamine oxidase, then the people tend to go for depression or suicide. If you identify them early, you can save them also. Genome editing, you know that it's challenging. We can change the defective gene with the normal now. So there are a lot of, I think I'll skip this, lot of challenges. When you do research on human subject, we have to have ethics. This is the kid here. The HeLa cells, I hope you know Henrietta Lacks, the cancerous lady, provided the cell, and her genome has been sequenced. The family members asking them, who permitted to sequence my aunt genome? So we have to take permission from individual and the family to work on the human system. Is very, very important for ethics. It has wide application. Genomics had wide application, genetic profiling, hybrid crops, livestock, how societal impact as man's own neo-eugenics, genomic revolutions created, new jobs, improve economy, comparative genomics help to understand the Nobel Prize, what they awarded him, and new microbes found in a gut system for our behavior is a new field from last 5 years also. And again, we have also contributed for that. And success story, understanding genomic variations, creating the vaccines, India become the world leader now. I hope you know that. Okay, understanding genomic variation, creating the vaccines for that. So to conclude, genomics is inspired thinking, it helps us to take the challenges, it affects the society, eliminate new hypotheses, and it transforms the way we understand many of the species that inhabit on our planet. It has wide application in almost all the fields, health, agriculture, fishery, forestry, environment, and ethics, and the social sciences. And the changing society to an era of personal medicine, whether you like it or not, already personalized, targeted, individual targeted, gene targeted medicine is available now. Another 5 years, you have to go for it. To get your own sequence, if you just pay 14,000, you can get your own exome sequencing and they analyze. You can have that as a like lipid profile. A day will come very soon. It will within 5 years. We think that we should go for it also. Even cost also is coming down. Even for targeted genome sequencing, if there's something suspected in your indication in your family, then you can go for target sequencing. Within 5,000, you can get those things and genomic variation identified. We can treat those kind of individual. So this is the group who the leader for the whole genomic analysis is Avinash, Kusuma, and Radhika, Raviraj, Megha, and these are my students. Even Swati, they work without other than the PhD for copy number variation. That has helped us to progress and contribute to the science. I'll stop it here. Thank you for your patience. I can have a couple of questions. And if you think the time is not permitting, in my first slide, I have my website or my nalurbr@gmail.com. You can send the question also. Now you can ask the questions. Thank you all.
Thank you, sir.
Thank you, sir.
Thank you, sir.
So any questions, suggestions, comments?
Hello, sir.
Yeah.
Stem cell therapy researches are going on regarding the non-verbal autistic student or autistic.
Yeah.
Hello? Can you hear me?
Yes. Tell me. Yes.
Stem cell therapy is going. I think in Bombay, I think it is mostly doing for the autistic students, which are non-verbal in nature.
Yeah.
Can you just can you elaborate what are the things they are doing for the stem cell therapy and for the speech introduction in autistic?
Yes, stem cell. So now my point is that I wanted to link the stem cell and the genomic variation. The stem cells, if it is normal, is not having any of now. The autistic, we also working on that now. Through PhD and now also we are working. Several genes are involved. Even gut microbiome also involved for the autistic. Depending on that family, all will not have the same kind of gene and mutation and so on. Whatever the autistic, the genes or genome variations are found in the Western data, it need not be in Indian data. So population specific, we have established that. So take the stem cells from their own parents who are normal, and those stem cells can be used for treatment of the autistic. And what kind of treatment, how one has to treat, that's also more important. So identifying the variations in the genes which are working on this, then we can also by using gut microbiome data, we can monitor giving right kind of the nutrients, right kind of the bacteria or the even the pill for that also. This is the kind of things are happening there. Yes, stem cell therapy certainly will going to be helpful, but it's a long way, but it becomes individual, family, and individual specific.
Can I, sir? Sir?
Yeah, sure.
Sir, thank you very much, sir. Thank you very much for a great presentation, sir. So I do have a couple of questions, like maybe a single question. So we were speaking about the height rate actually, sir.
Yeah.
So the first thing is that whether it can be called as a polygenic trait or a monogenic trait, because we call it as height and weight, but at the same time, when it comes to about lean and obese, we don't take it as a trait. Sometimes it is called as polygenic trait, but we always try to make it as an abnormal situation. But that same does not happen when it comes to short versus tall. Maybe whether it is going to be depending on how we are going to treat this or how people take this actually, because that's a great lot of difference. When we take high, small and tall as a trait, and why not a lean and obese as a trait? Kind of, we say them as abnormal, right? When he's obese, we call them as an abnormal person, but that does not keep when a person is short versus tall actually. Maybe you are you are the right person to ask that, so I'm just asking this.
Yeah, I mean, this is a common generic question people will ask, but the height and the short and the long the persons, again they have the limitation to certain kind of job there. Okay, the normal persons can do, the short person cannot able to. That's again the limitation. But that limitation may not affect the kind of things that takes place with the obese. Obese is also associated with other kind of the abnormalities and the metabolic activity, metabolic, the cellular functioning, it takes that. So coming to the genetic and environmental factor, both we have to consider that also. What kind of environment, if you provide, it can to some extent, it can modify our health. That's also we have to look into that. This other one is the genotyping here, a genetic problem. So now I'll take this diabetes. Earlier they called us only the insulin problem there. Okay, within the diabetes there are the MODI, we call them as a single gene defect for causing the diabetes, direct one. We can also identify them, and there are many genes present in this pathway. One family have different protein, another family have different protein also. We have to understand now diabetes become autoimmune diseases, and it depends on the environment also. So understanding more about the genetics and genetic factors is very, very important. And again, the right kind of environment one has to provide for them. This is more important. Okay, so again the kind of is it a monogenic or is a gene interaction, and what kind of combination that individual has, that's also more important. Sometimes only one or two factors is sufficient. Sometimes we do not know. We call them as the haplotype bin, 4 or 3 or 5 genes, the SMP contribute together to like for example, eukaryotic gene regulation, the transcription factors, they are also responsible for this. One had to go for complete genomic variation and look for this environment, the cellular environment and the environment, what kind of food, lifestyles are so important.
Yes, sir.